The state of Texas stands at the forefront of groundbreaking research and innovation, developing drugs that reverse the course of disease, putting technology in the hands of everyone, discovering novel methods to understand the unknown. The Academy of Medicine, Engineering, and Science of Texas honors those innovators who continue to advance the world around us. Established by Edith and Peter O'Donnell, these awards recognize the up-and-coming leaders of Texas innovation. Tonight, we present the 2008 O'Donnell Awards. Beth Levine has pioneered a completely new way of looking at the growth and deterioration of cells. This novel approach could unlock new information on the causes of a variety of diseases and how we treat them. Recruited to the University of Texas Southwestern Medical Center from Columbia University in 2004, Beth's research focuses on autophagy, the process where cells in the body literally eat parts of themselves. Autophagy is a normal, healthy function in cells, clearing the way for new cell growth. But when it's not working, the lack of autophagy can cause a myriad of problems.
Autophagy is important in preventing aging, it's important in protecting against cancer, it's important in protecting against many different kinds of infections, it's important in protecting against Alzheimer's disease and other neurodegenerative diseases.
Beth's discovery of the gene that stimulates autophagy, called BECLIN-1, has established a whole new area of research. Today, her lab is using simple models from worm, fish, and human cells, finding ways to turn on autophagy when it's not functioning properly.
Part of what she's going to do is to begin to take these lessons that have been learned in worms and flies and so forth and apply them into the clinical situation. I would say that's one of the most important things she's thinking about doing now.
The more that's known about this process, the more likely drugs can be developed to trigger autophagy in order to treat disease.
We hope to be able to develop new drugs that can be used to help treat patients with cancer and might be able to help treat patients with autoimmune diseases as well as with HIV and other viral infections.
Dr. Levine's research could one day open up a number of clinical fields that are relatively blocked off now, that we have no approach to solving them.
Beth is poised to take her research to the clinical level, where her determination for studying autophagy in the human body could provide whole new treatment options for patients.
I just really enjoy the process of scientific discovery for the sake of scientific discovery and the potential implications that this has for achieving a better understanding of how medically important diseases are caused and how we can develop better measures to treat those.
At Rice University, Jennifer West is known for two things: mentoring dozens of her students towards successful careers in bioengineering, and the progress she's made taking on some of medicine's most difficult challenges, like heart disease, stroke, and cancer.
We're trying to tackle some of the biggest problems where there's a lot of room for improvement, and we're trying to take some really new and different approaches to these problems.
Jennifer grew up in a family of educators. Her early interest in science and math led to a doctorate from the University of Texas at Austin. Today, Jennifer's lab at Rice centers much of its efforts on developing new materials for surgeries related to coronary artery disease, an ailment affecting more than 14 million Americans. Jennifer has developed a synthetic material that, when combined with a tissue sample, can grow into a new blood vessel. This could potentially greatly simplify coronary bypass procedures and reduce complications.
From harvesting the tissue to having something ready to go back as an implant would take 14 to 21 days. So it wouldn't be something available for emergency use, but when you have time to plan the procedure, it would be a definite option.
Jennifer's also applied her research to enhancing stents used in balloon angioplasty surgery. A drug-releasing coating for stents developed in her lab can help eliminate the risk of scar tissue buildup and additional blockage.
I think she just is a person who not only has extraordinarily creative scientific ideas, but can translate those into new technologies that have the potential to help patients.
Jennifer's lab is also working on new therapies to treat cancer. She's using nanoparticles that, when injected into the body, attach to cancerous tissue. When exposed to infrared light that's harmless to normal tissue, the nanoparticles heat up within the cancerous tumor, killing the unwanted tissue.
Her group has been involved in very successful animal trials to test that technology, and they're preparing for the first clinical trials in patients.
During my career, I've had several things go all the way from initial basic research to things used to treat humans. That's really what it's all about. You really want to see the research impact people's lives, change and improve their lives.
Even with so much success in the lab, it's the accomplishments of her students that are most fulfilling.
I think actually seeing the young people who I've helped train come out and be really successful has been my proudest accomplishment.
Most all research demands extreme patience. Breakthroughs take time. But when it comes to learning more about genetics, Edward Marcotte was determined to speed up the process.
Part of what we do is we try to develop technologies that allow us to make measurements on thousands of genes at once. So we end up doing things like roboticizing them so that you can do a bunch at once.
As a UT Austin undergraduate, he got his first exposure to studying genetics.
Partway through college, I ended up taking a class in genetics, and I was just really amazed. It was fascinating to learn about what was known about biology, how your cells worked. And it's just absolutely amazing.
After earning his PhD at the University of Texas, he spent 4 years at UCLA for his postdoctoral studies before returning to UT Austin and starting a lab in functional genomics and bioinformatics. Edward's lab enlists computer scientists, engineers, molecular biologists, and chemists. This interdisciplinary approach has produced the most complete research available today for how proteins within cells function. This new research is fundamental in understanding what different genes do.
One of the most important issues in science is asking questions and asking the right questions. And the second, perhaps most important issue, is being able to develop a plan to implement the quest for addressing these questions. And he does both.
To increase the pace of his research, Edward's lab has developed technologies enabling them to experiment on thousands of proteins and genes at a time. Working with basic genetic structures in yeast and tiny worms, they're discovering possible correlations to human genetics.
More recently, he identified a number of different genes that can cause cancers in both model organisms and also very likely in humans.
We've looked at things like predicting which genes make worms live longer if you target them with drugs. And we were able to successfully predict genes that would increase the lifespan of a worm by 50%. Again, it's not a human, it's a far cry, but humans have those genes. And so it's possible that the same thing would work in a human.
As Edward's team characterizes the function of previously unknown proteins and genes, they are better equipped to predict what happens when genes malfunction.
What I hope is that some of the things that we're developing would translate into being able to diagnose genetic cause of disease and actually be useful in that way.
By emphasizing new techniques as much as his ongoing research, Edward is substantially advancing our knowledge of genetics.
It's very satisfying to think that you could exploit your curiosity to actually do something useful.
For Samir Pendarkar, the future of technology is right around the corner. With 30+ patents to his name, he's become one of the leading engineers at Texas Instruments in developing the technology powering the consumer electronics of tomorrow. Think cell phones that rarely need charging and cars that drive themselves.
We can come up with products which will really make a difference in the sense that we can have, like, say, for example, a cell phone where we can integrate different things like GPS and TV and radio and things like that, and you just make them more mobile, less dependent on battery.
Born in India as the son of an engineer, Samir grew up fascinated with electronics.
I always used to tinker with electronic things back at home, and that kind of just fascinated me.
Today, Samir focuses his research on how low-voltage electricity travels across tiny transistors and chips. His research uses advances in semiconductors that make it possible to do much more with less power.
The circuits that he's designed and the processes that he's developed are used to make chips. And if you've ridden a car, an automobile, or used an electric toothbrush or a shaver or a battery charger for a cell phone, it's very probable that you've used one of the chips that Samir has helped develop and manufacture.
The microchips Samir and his colleagues at TI work with today are only a fraction of the size of those from just 2 years ago.
So these are essentially what the chips look like. As you can see, the size of this is amazingly small. I mean, it's smaller than a peppercorn, if you will. It's just getting smaller and smaller.
Samir envisions a time when these advances will result in electronics that make our world safer and smarter for everyone. As a result of Samir's work, safety devices only found in high-end cars like tire sensors and anti-locking braking systems will soon be available in all new cars. Samir's research will help make this technology more affordable so more people can benefit.
It is not every day that one comes across a talented individual or an engineer that has contributed so much in such a short time. And Samir has managed to accomplish that.
The knowledge that what you're working here is going to go into a product which is going to basically make a huge difference in the society as such, just make it better and safer and just have more people access to these basic things like safety, which I think is one of the things that I hold very dear.
Tonight, the Academy of Medicine, Engineering, and Science of Texas congratulates Beth Levine, Jennifer West, Ed Marcotte, and Samir Pendarkar, the recipients of the 2008 Edith and Peter O'Donnell Award.